Well, now for something completely different. And I'd like to start by thanking the organizers so much for inviting me to contribute to this tremendous meeting. I think we'll be starting in a moment with, I'm going to start with a textbook explanation for why insects are so fantastic, and so successful, and I agree with everything that's written here, and it's been illustrated by a number of the talks we've already heard. Their diversity in terms of species and form and function, some of their behavioral and social complexity, and their importance in terrestrial ecosystems. But there's another factor that's really important that at first sight looks like a real limitation. That is that these insects, like all other animals, are metabolically impoverished, just like you and me. So insects and we can't synthesize 9 or 10 of the amino acids that make our protein, essential amino acids. We also can't make coenzymes absolutely required for the normal functioning of enzymes central to our metabolism, i.e. the B vitamins, and insects additionally can't make sterols, which are really essential for the very architecture of membranes. And therefore, we would expect insects would not be able to utilize various kinds of diets. In particular, vertebrate blood, which is grossly deficient in B vitamins, plant sap, grossly deficient in essential amino acids, and here I mean both the phloem sap, which mediates the transport of photosynthetic sugars, and xylem sap, which mediates the transport of water in plants, and then various organisms that utilize sound wood, whether in the forest or in your furniture, as well as various stored products, which are deficient in a wide range of these essential nutrients. And in this respect, we could, as you might say, insects haven't read the basic nutrition textbooks because there are plenty of insects that utilize these food sources through the life cycle. For example, in relation to vertebrate blood, the bed bugs among the hemipterans, all of the sucking lice, and various true flies, such as the tsetse fly, feed on vertebrate blood grossly deficient in B vitamins right through the life cycle. Similarly, feeding on plant sap has evolved multiple times within the hemiptera, although notice nowhere else in the entire animal kingdom. Hemiptera are wonderful. And the utilization of sound wood and co by particularly a number of beetles, including bostrichids and anobids and some weevils and so on. Now, these insects appear metabolically to be doing the equivalent of walking on water. That is, they're doing something that cannot be achieved. But of course, they do it because they've entered into alliances with microorganisms that function as the nutrient factories of my title. These microorganisms specifically overproduce nutrients in short dietary supply. I'm going to be talking about plant sap-feeding insects because they're the ones we work on. And the phloem and xylem sap of plants is grossly deficient in essential amino acids relative to the protein of the insects. And the bulk protein of these insects is just like that of all other animals, roughly 50% essential amino acids. And their microbial symbionts release between 50 and 90% of the essential amino acids that they make. Your average human biotechnologist would give their eye teeth to have access to these types of microorganisms with this sort of capability. And these microorganisms enable these insects to grow and reproduce. So here's a couple of examples of these insects in whiteflies and in aphids. And here there's a piece of vocabulary you'll need to know for the next 15 or 20 minutes, the word bacteriocyte. And that is a very specialized insect cell whose function is to house and maintain these bacteria. For example, in this lovely fish micrograph, I hope you can see it with the lights, these cells lit up in red, which is a fish micrograph of the bacteria, Portiera, present in these cells in the body cavity, in the bacteriocytes in the body cavity of whiteflies. And this nice micrograph of Simon Chandler's showing the Buchnerer bacteria of aphids in the cytoplasm of these bacteriocytes, again in the body cavity. So if you'd like to think of your liver in your body, well, these insects, the bacteriocytes of these insects are equivalent by biomass to our liver, the difference, of course, being that the cells of these bacteriocytes are full of bacteria. Now, these bacteria are with these insects always because they're transmitted vertically from mother to offspring via the egg, and indeed, all the indications are that they've been doing this without fail in both of these two groups for at least 100 million years. As a consequence of this, there's relaxed selection on genes which aren't required in the insect, genes only required in their ancestral free-living condition, and also because very small numbers of bacteria are transferred from mother to offspring, there's a small effective population size, and so we get genetic drift. And so you can see the genome size of these bacteria is really small. So what I'd like to do today is just tell you some work that we've been doing using metabolic models to investigate these overproducing microbial nutrient factories. I'm going to start off with a genome scale model of the individual bacteria, showing that actually these metabolic networks are very fragile, and then go on to some multi-compartment models investigating what's metabolically feasible and also the costs of maintaining these bacteria. The basic idea is very simple, and this idea I developed in collaboration with my very good colleague Gavin Thomas, who knows nothing about insects. He's an E. coli biotechnologist, really. And so what we did was take the genome, annotated genome, of the Buchnerer bacteria in the pea aphid, identified all the metabolism genes, and then we, from that, inferred the number of reactions and number of metabolites. And then we reconstructed the metabolic network, and using flux balance analysis and related techniques, we estimated the flux through that metabolic network according to some desired outcome, and the outcome, generally, most of our in silico experiments, we use biomass production. We've done a number of experiments in silico, and I think the following one is quite informative. Sorry. There we go. So in this experiment, we individually deleted each metabolism gene and then scored the in silico growth of the bacteria. And as you can see, the great majority of these metabolism genes are individually required, more than 80%. This is really fragile. These Buchnerer certainly haven't read the nutritional textbooks, which tell us that metabolic networks are really robust. For E. coli, or for you and me, the number of essential genes would be the order of 10 to 15%. For the Portiera in whiteflies, and this is more recent work we've done headed by Nana in the lab, the number of required metabolism genes is even higher at more than 90% of the total. In addition to being metabolically very fragile, it's very interdependent with the host. And this is indicated by the number of what we call mobile metabolites, that is being transferred in one direction or another with the insect host. And as you can see, it represents nearly a quarter of the Buchnerer metabolites for the aphid, and just coming up to half for Portiera. And it didn't have to be like this. It would be perfectly easy to envisage a very small number of metabolites being as inputs into the metabolic network of these bacteria, and just each of those 10 essential amino acids as outputs. If you don't enjoy metabolism, you might consider this as the equivalent of being like a metabolic chatterbox. They're interacting in lots of different ways metabolically with the host. And a major part of that is shared metabolic pathways. Here's an example of a shared metabolic pathway that has evolved independently in the aphid Buchner system and in the whitefly Portiera system. And it relates to the biosynthesis of branched chain amino acids, isoleucine, leucine, and lysine, isoleucine, leucine, and valine, which are also essential amino acids. And as you can see, independently, the Buchner and the Portiera, the symbionts in each of these two insect groups, have lost the gene mediating that terminal transamination reaction. But if you, and we have done, determine the transcriptome of the bacteriocyte, that is the insect cell containing these bacteria, we find that it's highly enriched in an insect enzyme, the branched chain amino acid aminotransferase, or BCAT for short, which is an enzyme that's found in most animals, including ourselves. And this enzyme generally mediates the first step in the degradation of branched chain amino acids, but it's reversible, and it can also function in the biosynthetic direction. And we've been able to demonstrate empirically by metabolism-related experiments that, in fact, this is how it functions in the bacteriocyte of the aphid, and we anticipate it does the same in the whitefly Portiera system. So here's an example of a shared metabolic pathway. Another example, and this is a little bit more complicated, and Jumbo Luan in the lab has done a lot of the work involved in this. So here's our Portiera within this box, and this is the lysine biosynthetic pathway. And you can see immediately that it's missing the two terminal reactions in lysine biosynthesis. And in addition to that, one of the intermediate reactions, the gene DAP-B, appears to be a pseudogene. And we certainly get no evidence of any production of protein. And as you can see, Jumbo conducted a transcriptome analysis of the bacteriocyte, the cell containing the Portiera, and found it highly enriched in precisely the transcripts for precisely the genes that are absent from the Portiera. Now, this was really most exceptional because these are not genes that you find in animals. They're bacterial genes, and Jumbo was able to go on to show that not only have these genes been transferred, they've been transferred directly to the whitefly genome, and that they're of divergent bacterial origin, none of them related to the Portiera symbiont. Quite clearly, if we're going to understand the metabolic function of these Portiera, we can't do it on their own. We have to do it in conjunction with analysis of the insect cell using the transcriptome as a basis to reconstruct the metabolic network for that bacteriocyte. At this point, I have to be like the British civil servant. I've been a little economical with the truth. Because in my case, I have missed out another partner in this association. So there's another bacterium in the bacteriocyte of the whitefly, and you can see this in this lovely micrograph of Hongwei's, the central lobed nucleus in pink, the Portiera, which I've been talking about up till now, and then the second bacterium, Hamiltonella. So here we have a three-compartment network. And we've analyzed this network, taking account the difference in biomass abundance of the Portiera and the Hamiltonella. And that network, well, it looks a little bit like a hairball. It's an awful lot more complex than those single genome networks of the individual bacteria. Indeed, we had to call in reinforcements because our life was really getting computationally rather demanding, and this work has been done in collaboration with Brandon Barker at the Cornell Center for Advanced Computing. So we've used this multi-compartment metabolic network to investigate a number of different things, but I'd like to focus on just one, and that is an aspect of the biology of Hamiltonella. This bacterium has the genetic capacity to synthesize two of the essential amino acids, lysine and threonine. And quite rationally, our colleagues who initially sequenced this Hamiltonella genome inferred from this that they are overproducing these two amino acids and making it available to the insect as a supplementary source of these two essential amino acids. So we investigated that, making use of our multi-compartment network. And here is the data for the Hamilton, this little portion of the Hamiltonella network. And you can see they're both derived from aspartate, and you can see that in our metabolic models, they very readily make the two essential amino acids, but they invariably allocate those amino acids to protein. Whatever we did, we couldn't persuade our metabolic model to do what our colleagues said happened, which was to overproduce, except under one set of conditions. And that was when we applied an unlimited amount of exogenous aspartate substrate. And under those conditions, we could persuade our Hamiltonella in silico to overproduce lysine, but even under those conditions, not threonine. I've reproduced these data here, just precisely here, and added in the Portiera data. Portiera can't make aspartate at all, so it's always exogenous, and we obtain very high rates of overproduction and predicted release to the host. These data are in very large part very compatible with data we've collated both in silico and empirically, that is, doing real metabolism experiments at the lab bench, which indicates that the host has very tight control over nutrient production by supplying the substrate, in this case aspartate. That is, we call it push metabolism, meaning that the production of essential amino acids by these various intracellular bacteria is mediated by the amount of substrate supplied from the host. That these bacteria are poised for some sort of maximal rate of essential amino acid overproduction, and the realized rate is determined by how much substrate comes in from the host. In other words, we have a complete restructuring of regulation of nutrient production from what we find in free-living bacteria, where the bacteria produce as much as is needed. You get product inhibition. There's no product inhibition here. It's regulated entirely by the host, with the opportunity of really precise control. I'd like now very briefly to move on to the last part of my talk, and this relates to the cost of maintaining these bacteria. We've been studying this in the phloem sap feeding insects, but I'm going to talk here about our work on xylem sap feeders, because xylem sap, remember, is what transports the water in the plant, and it is incredibly nutrient poor. And therefore, we would expect these insects to be under enormous selection pressure to be as efficient as possible in the nutrients that they acquire. So we've been studying three groups of xylem feeders, the spittlebugs, the cicadas, and the sharpshooters. They're all members of the suborder Orcanorhyncha within the Hemiptera, and they're all derived from an association in the ancestral Orcanorhynchon with a bacterium called Solsia, which is a Bacteroidetes, which we believe was ancestrally associated with another bacterium. Now, Solsia has been retained by all the xylem-feeding Orcanorhynchons, and as you can see, it has a really tiny genome, but there is different, we call it a companion symbiont, in each of the three different groups, as indicated here, the Sedalis, Hodgkinia, and Baumannii. Now, we know from genomic data that the Solsia can synthesize and therefore presumably release either 7 or 8 of these essential amino acids, and the various different companion symbionts can synthesize and presumably release the precise complement of those essential amino acids. And here we have essentially the same structure of a metabolic network, of a 3-compartment network between the Solsia, the companion symbiont, and the bacteriocyte. So we conducted this, and this is the work of Nana and Besam Choi, who's also involved in this work. So we reconstructed these 3-compartment metabolic networks. Actually, I make that sound so easy, don't I? It was an enormous amount of work. And I'm delighted to say that all of the bacteria overproduced in the way that was predicted from the genome, really different from the Hamiltonella. Now, at first sight, all of this looks like wonderful evolutionary design with perfect complementarity of essential amino acids and so on. But if you think about it a little harder, it becomes immediately obvious that there's something doesn't quite add up here. Here we have hosts that are bearing the costs, presumably, of two different symbionts which mediate the same amount of functions as a single symbiont in other associations. And this was why we were particularly interested to explore the costs of these xylem-feeding insects, not just because of the diet, but also because of the way the symbiosis is organized. We used two ways to investigate the cost. Our first way was to quantify the efficiency of nitrogen conversion to essential amino acids. So how much nitrogen do these bacteria consume for giving the amount of essential amino acids back to the host, reminding totally non-metabolism people that essential amino acids have lots of nitrogen in them. Well, the Saucia are really efficient. They have an efficiency of about 50%, which is about as good as it gets. But our companion symbionts really are not playing the game, and that applies particularly to the Baumannii in the sharpshooter, where the conversion efficiency is less than 10%. This is costly. We then went on to actually, costly with respect to nitrogen, we then went on to consider the total cost, and the result is, we were actually really surprised by this result, but it's very robust, that according to the genome size, the cost declines with declining total cost of maintaining symbionts, declines in a very uniform manner with actually the size of the genome. So in other words, the cost of maintaining a pair of symbionts is compounded by the inefficiency of nitrogen transformations in the companion symbionts, but is ameliorated by extreme genome reduction. So I'm now moving to my penultimate slide, which is my conclusions. I hope I've been able to show you that microbial symbionts are tremendous nutrient factories, overproducing, in this case, essential amino acids, but they're metabolically very fragile, and also they're really quite costly. I think this is actually a really nice example of metabolic coevolution between insects on nutritionally inadequate diets and intracellular symbionts. And I'd just like to finish off with a few acknowledgements. We can't do anything without funding. We are so grateful, particularly in relation to this work, for funding from NSF, the Cassava Whitefly project, in relation to, funded by the Gates Foundation, and also the Aristotle Cloud Foundation. And I've actually put everyone on here, most of the past, all of the present, including our collaborators, because surely everything is connected, certainly is in our lab. But the special heroes of today are, of course, Nana and Bessem and Jumbo, together with Brandon and, of course, Gavin. Thank you very much, everyone.
Thank you. Thank you. I'm sure there are a lot of questions. So, there. Take a microphone. Yeah.
Thank you for this very interesting talk. I was wondering, did you find species with only Saucia as a symbiont and no companion symbiont to compare whether it would be a competition effect with the companion in producing and converting?
Yes. We have never found a xylem-feeding species with Saucia only. There are one or two reports in the literature, and I think they do need to be investigated, perhaps a little more carefully. But we have, there is one very interesting genus of spittlebug, which has three partners. It has the Saucia, the ancestral beta proteobacterial symbiont, and the replacing Sedalis one. And we're starting work on that to actually come to understand how the replacement may be happening. Yeah.
Thank you.
Another question there?
Yeah, hello. I was just wondering what happens to the intracellular symbionts when they die. Is there sort of some nutrient recycling from those cells?
When individual bacteria die, yes. The very few of the bacteria survive, die right up until mid-reproductive or beyond stage of the insect, and then there is very substantial cell death. The relationship does appear to be biotropic rather than, they're not, the insect isn't eating the goose that lays the golden egg. Yeah.
Last question before lunch.
And how's the bacteria transmitted to the progeny? So is it found in the germline or inside the egg?
Yes, so the bacteriocyte is a somatic cell, and the bacteria are released from the bacteriocyte, and then they have a relatively fleeting free phase within the insect tissues, and then they're taken up by the oocyte in the ovary, okay, and then become distributed to the bacteriocyte that develops in the embryo.